As a physician, you want to offer your patients the best treatment available. Taking biopsies and screening suspicious tissue for cancer tumours is becoming increasingly important in clinical practice.
For us it is very important to have the right diagnosis of a patient. Generally we do this by taking a biopsy and for example in a couple of patients we know that these biopsies are not representative. In lung cancer actually 25% of the biopsies are not representative.
Physicians are severely hampered by the lack of precision of the needle tip location during an intervention. The patient sometimes needs to undergo a second biopsy, with again the risk of complications and the delay for treatment.
It is very important to have a biopsy needle which can sense the tissue at the time of the biopsy.
Real-time characterization feedback from the tip of the smart biopsy needle can significantly reduce false negatives and improve the accuracy of diagnoses.
This is a spectral tissue sensing system, which is a box indicated here with a light source. And that light is connected via fibers to the end of a needle tip. You shine light into tissue and light is scattered and absorbed, and part of the scattered light is then detected via a second fiber integrated in the needle. And what the device does is translate that into a spectrum displayed on the screen, which can be identified and which can be seen in real time by the physician.
Spectral tissue sensing using photonic needles has the promise to be a valuable tool for screening tumours. However, the cost and size of the spectrometer console needs to be improved dramatically in order to make it commercially viable. With the support of the European programme Horizon 2020, together with 8 partners from industry and academic teams, the project InSPECT aims to develop photonic light sources and miniaturise spectrometers for real-time and low-cost tissue sensing.
The project aims at realising a LED-based compact light source, a cubic inch size compact microspectrometer and a spectrometer device on a photonic chip.
Miniaturized devices such as the cube or chips can be replicated at a lower cost to make them more accessible for widespread use, such as at physicians' consultation rooms and decentralized health centers. Ultimately, these photonic spectrometers open the door to new surgical devices.
Actually, we would like to use these sensors in smart surgical devices so that the surgeon exactly knows whether he's cutting into tumor tissue or into normal tissue. Any other application would be, for example, to use these sensors in implantable tissue sensors.
The InSPECT research can also be used in other domains, such as personal fitness.
Our ultimate dream is to have photonic spectrometric devices available that can be integrated into a watch or into a mobile device, for instance, to monitor personal health.